Is it on? Okay, good. Thanks, Interspecies and CUK for giving me this opportunity to visit your campus. This is the first time I'm coming here. I'm really, really impressed by, the place.
Arnab, can I just interrupt for 1 minute? Yeah.
Are you going to introduce me?
Yeah, but their introduction slide was not there. Here it is, something is there. And I know him very well. So I cannot stop my temptation for introducing him. He is a brilliant scientist. He is a Jesse Bose Fellow. He is the elected Fellow of National INSA. He is a Star Awardee and many GRC. Okay, yes. So these are also everybody I told. He got the National Bioscience Award, Among Swami Fellowship, and many more. Also Nasi Fellow. So with this, yeah.
Thank you.
Large number of people, has good publication, yeah.
I thank him, and again, thank you guys for giving this opportunity to come and speak to you. So my lab is fundamentally interested in understanding the molecular basis of aging, and if you change it, I'll put my real stuff. Yeah, so some of you may be wondering what my title means to you. The title has to come. It's called Interspecies Interactions That Ensure Optimal Lifespan. So does it make sense to you? It took a long time for me to make sense of it, but and I think the computer is also not liking the title. So today, what I'm going to tell you is, if you consider. Okay, cool. So let me first do the necessary introduction. It's fine, I'll take care of it, don't worry. So I come from this institute. It's called National Institute of Immunology. It's in Delhi. It's a premier institute of the Department of Biotechnology. Now we have its display settings. Okay. Why does it not? Okay, cool. So this is a premier institute of the Department of Biotechnology, and we work on many areas of biology, including immunology and infectious disease, and of course cancer biology and non-communicable diseases. And we have PhD program. We have integrated PhD program through a consortium where you can work with many labs of the BRIC laboratory, which is our parent body. And importantly, we have very good master's training programs. If you are interested, we have 6 months to a year of master's training, and we also take undergrads who come over for the summer for 2 or 3 months, and they work in our lab. So it's a great opportunity not only to do your PhD, but also to engage with us in different times of your career. So. I started by saying that, what's that fancy title? So the title means that I'm going to talk about the interaction of bacteria that infests our body and the host, that is us, and how they communicate with each other and fight it out. Okay, so this is not an infectious biology talk, which you might be expecting. It's a harmonious coexistence of bacteria with the host and how we thrive and why each of these two organisms and their interactions are important for optimal lifespan. So, but I'll start with this very interesting question that which I typically, when I go to undergrad colleges, I tell why do you use model systems in biology? So, had it been like a very two-way communication, I would have asked you for the answer, but I'll just give it. So life is complicated, right? So you all understand going through a lot during your master's and PhD. Biology is even more complicated because now you are expected to find mechanisms and publish papers. So as many of my colleagues talked about human biology, it's often very difficult to understand mechanisms in those contexts. So there is a need for organisms which are easy to tame. Tame in the sense that not like taming a lion, taming them genetically, right? So to modulate their genome, to understand biology through tinkering with their genes. And very early in history, not too early, but very early in history, these two gentlemen, I understood the importance of model organisms, and they all knew about biochemistry. They know, then that time was biophysics was also developing, but they understood that the complete biology cannot be understood inside a tube. So they proposed beautiful model systems which we all have studied in our developmental biology courses, and Morgan and Sidney Brenner, much more recently than Morgan. And they introduced two of the greatest model systems that we typically use in our labs. And one is Drosophila, the other one is C. elegans. And I'm going to talk today about how we use C. elegans to understand the complex biology of aging. And it's to study biology of aging, there are many model systems. You know many of them, and you will be surprised that it starts with yeast. You can understand, and many of our fundamental understanding of the biology of aging and why we age and whether we can delay the onset of aging comes from this single cellular eukaryote. You also have Drosophila, one of the very important model system, mouse. Some zebrafish models are used for aging research, although they are very long-lived. And very recently, though, there is an African killifish which is now being touted as one of the best models of vertebrate aging because it lives only for 3 months. And of course, you need mouse, which lives for many years, and you don't want, as a PhD student, to work on mouse aging. It's very difficult. So in our lab, we just chose the easiest of them all. We use C. elegans as a model system, and I'm going to briefly introduce you to C. elegans. If you haven't seen them in your laboratory, you can always come by and to my laboratory, or there are many laboratories around this area which works with C. elegans, and you can take a sneak peek at their life history. So this is how they look on the plate. They are a millimeter long. And they go through many developmental stages, which I'm not going to talk because of the paucity of time. But fundamentally, it's a very cheap model system to use in the laboratory. So you can see that it has a short lifespan. That's absolutely what matters to my PhD students. Okay, they don't have to worry too much. They live only for 25 days with an average lifespan of around 17 days, which is absolutely invariant. Whether you do it in CUK or you did it in Delhi, you'll get the same lifespan of average 17 days. So it's highly constant, okay? It is very cheap to maintain. You can maintain them by, growing them on agar seeded with bacteria. It's bacteriovore, it eats bacteria. It's very easy to store. You can simply freeze them and put it in a -80 freezer, and 20 years later, you can come back and thaw it. They will go through the entire life cycle as if nothing has happened. They are transparent, so you can use all kinds of microscopy, and I'm going to show you a lot of images of mitochondria, how we look at them in the live worms, and is backed by powerful genetics, and is amenable to very many high throughput techniques which we are using for drug discovery purposes. 70% of his genome is conserved, so whatever you are finding in C. elegans, many of the times they are conserved in humans. So it gives you a very delicate balance with ease of genetic manipulation on one hand and relevance to human. So it's a model system of choice. And if you belong to a small college in some remote part of the country, you can still use C. elegans, unlike Drosophila or any other systems, because this is all you need to run a basic genetics lab of Drosophila. And trust me, many of the fundamental discoveries which people have got Nobel Prize for, were done using this set of instruments. Okay, so all you need are Petri plates, an autoclave to make media, a stereozoom microscope, a couple of incubators to store the worms, and the most inexpensive but the most important step is a glass rod fitted with a platinum wire, with which you can delicately pick the worms and manipulate them. So if you look at the worm anatomy, it's nothing but a hollow tube filled with stuff which often represents the organs of our body. For example, it has an elaborate digestive system and an elaborate reproductive system. So it has two gonad arms, forward and back. And in middle, it stores its eggs. And you can see lines of oocytes when these worms become adults. If you cut sections through the worms, you can see that there are the dorsal nerve cord, the ventral nerve cord. It has 302 neurons. And this is the intestine, and these are the gonad arms. So it's a very simple anatomical feature, and many of these biochemically represent what is going on in our system. The complete somatic lineage of these worms are traced. And the advantage for it means that you know the antecedents of each and every cell of the 1,059 cells in the body of these worms, and when and where they originated. So very early on, researchers who did not have these fancy molecular biology tools and wanted to study a particular neuron would go and use laser to ablate one of the lineages and stop having one kind of neurons or the other. So using this very simple technique, they could map out many disease genes, and that formed the basis of using C. elegans in disease biology. As I told you, C. elegans is absolutely transparent, so you can use C. elegans for many biology which has to be visualized. And I'm going to show you one, for example, where you can look at early embryogenesis. You can just simply focus your microscope and your camera on a single oocyte which is fertilized, and see it going through all the developmental stages and look at the time. So if you have a time lapse photography of 6 to 8 hours, you start seeing the entire worm developing just before your eyes. If you are a molecular biologist and you want to study gene expression, for example, you can tag the histones with different fluorophores, and you can fancy yourself watching cell division in multicolored, multi-dimensional, three-dimensional, whatever. So, and if you are a fun-loving person, like some of my students do, and they're interested in neurobiology, and they use the worm's inherent capability to sniff out chemicals which are sweet-smelling, and oops, it doesn't like this. Okay, so yeah, so you can actually use the worm's ability to sniff out chemicals and put them at appropriate positions in the plate and put a little azide so the worms get paralyzed and you can end up having a smiley face. So this is simply not exciting undergrad program, but the important part which I want to tell you is that we know everything about its neurons. There are 302 neurons, and this is one of the only model organisms where you know the connectome. You know that how each and every neuron and interneuron connect to each other in the whole organism. And you will be surprised that now we know how a touch response sequentially fires this network in order to move a muscle. So that is the kind of resolution of understanding that we have of how the worms can respond to different biological cues. So the worms can also be used for disease modeling, as I wanted to just show you an example. And this is a disease modeling of axonal injury, and where using you can use either either laser, or you can simply twist the worms to break the axon, and then you can follow through in a time-lapse manner and see that the axons regenerate and connect. And if you have mutants in certain pathways, including the Wnt signaling pathway, these worms cannot now get back their connections. And what are the consequences? You can use a simple physiological assay of touch response, where you can touch these worms with an eyelash, and you see this sinusoidal curve coming back. If any of the genes which are important for axonal regeneration is perturbed by either by GNA mutation or by RNAi, you would not see this kind of backtracking. And you can automate your microscopic setup in order to do large-scale screens to identify mutants which promote or demote axon injury repair. So this is how powerful this model system is. And so when you have such a powerful model system, you try to ask difficult questions, right? So using this model system, we started asking, why do we age and can we prevent it? When we say we, it doesn't mean me, because there were a lot of people before me who already asked this question, and I joined the bandwagon to understand some part of it. Now when you look at our current understanding of aging research, this is so new that it has not yet reached your textbook. I'm sure none of you have seen this primary diagram in any of your textbook. But if you read this paper in Cell in 2023, and this is a series of paper where the scientists are trying to put together the hallmarks of the reasons why we age into a model, and this comes out every 10 years in Cell. You will see that there are primary causes of aging, which is, you've heard about genomic instability, telomere attrition, loss of protein folding and degradation machinery, disabled autophagy, which is, the recycling of your cellular organelles and proteins. So these are all primary causes of aging. They lead to aging directly. There are some antagonistic causes of aging, which are important developmental processes, which, because there is no selection for organisms to live longer, these processes get deregulated with age. These include nutrient sensing. This is important to know, that what you eat determines how long you will live. So nutrients regulate longevity. Mitochondrial dysfunction. Mitochondria are important regulators during your development. You need a lot of power in order to divide and differentiate. But when you become older, this becomes a burden. So it's an antagonistic cause of aging. And of course, you have all heard about cellular senescence. Now there are integrative causes of aging, which happens because of these two primary and antagonistic causes. And the primary, most important, is dysbiosis. And I'm going to talk about dysbiosis today. There are chronic inflammation, altered intercellular communication, stem cell exhaustion. So these are the 12 hallmarks of aging that we currently know of. New things may be added when we read the review in 2033, and you could be part of that journey. So when I was a postdoc, I jumped from plant sciences to animal sciences because I was fascinated by the biology of aging. And over this last 20 odd years, me and my very, very capable and wonderful collaborators, we are trying to contribute to the hallmarks of aging and of course hallmarks of cancer. And today I'm going to tether together a story which connects dysbiosis, which is the deregulation of the interaction between the gut microbiome, which many of you have heard about this term, disabled autophagy, deregulated nutrient sensing pathway, mitochondrial dysfunction, and altered intercellular communication. So a lot of things to talk about, but I'm going to try and simplify that as much as possible. So let us go back to what is important. So I don't know how many of you have heard this word microbiome, but this is like the genes that Dr. Saab had shown you. This is the current fad in biology to understand how microbiome regulates many aspects of disease and life. So you are probably having more number of bacterial cells in your body than the total number of cells that make you human. So you are more bacteria than you are human. That's because there are 10x more number of cells, bacterial cells, inhabiting your gut compared to the number of cells that constitute a human body. So while you can code for 22,000 genes, the bacteria in your gut provides you 3.3 million non-redundant genes. So they are additional coding power. They empower you, right, to code for things that you are not capable, you do not have the biosynthetic pathways to code for those genes. And importantly, while in this room, if you sequence your genomes, 99.99% of your genomes will be same, but only somewhere between 80 to 90% of your microbiome will match each other. So you distinguish yourself by having a different microbiome, because that's determined by what food you are eating, what is your genetic susceptibility, and so on and so forth. So what I told you is the microbiota, or the bacteria, as well as fungi and phage that infests your body, both on your skin and in your digestive tract, mainly in your intestine, provide supplementary genome. They often explain some of the missing heritability traits. Very important source of energy, because they can produce short chain fatty acids, which your cells can use as energy. It trains your immunity. But for today's talk, you have to remember that they provide vitamin B12. So none of the organisms can make vitamin B12 except some bacterial species, and we directly or indirectly derive vitamin B12 from them through diet, because of our association, or when your doctor prescribes you those pills. So if microbiome is such an important asset of your body, if there is dysbiosis or disbalance in the bacterial load that is existing in your body, you would have diseases, right? Very importantly, most of the diseases of human are attributed or can be correlated to changes in the microbial diversity. But most interestingly, if you see, most of the diseases that are listed here are age-related diseases, right? You see Alzheimer's, Parkinson's, anxiety, depression, obesity, cardiovascular disease, diabetes, insulin resistance, all of them are age-related diseases. So this tells us that the microbiome or the microbial diversity in your body will determine how fast or slow you are going to age. So how do we study this complex interaction between bacteria and the human body, right? You need model systems for that. You cannot possibly do mechanistic studies in humans, but it is very difficult to study it, because you will not see differences in phenotypes simply by changing diet, right? If today you are eating, one kind of food, and Indrashish has made so great arrangements. Every day you are eating different food. Does it change your life history trait that quickly? No, because your body has intrinsic mechanisms to prevent diet or microbiota from changing your metabolism. Okay, so there are genetics of your body preventing microbial or diet-induced changes from affecting your physiology. So that's why you do not see those differences. When do you see that? When you have a risk allele. For example, if you have a risk allele in the fat mass and obesity gene with an AA genotype, you will have visceral obesity even if you are having normal diet. Similarly, if you have a risk allele in the BHMT gene, you will develop cardiovascular disease even if you have normal diet. However, if you now change your diet to include vitamin B12, cobalamin, folate, which often comes from the bacteria, your microbiota, you can change the risk, or you can prevent the occurrence of this disease. Telling us again that this intense interaction between diet, microbiota and your genetics determine your rate of aging and the incidences of age-related diseases. So in real life, if you want to understand aging, you have to understand the interaction between diet, microbiota and your genes, and which together we call a trifecta. So it's easier said by done. You know that this is so complex biology. So we go back to C. elegans as a model system. And today I'm going to tell you one story that we are, we have communicated. It's unpublished still. It's on bioRxiv, where we use C. elegans to decipher how vitamin B12, which comes from the microbiota, influences aging, and what are the genes in the body of the worm, and also there in your body, which prevent this physiological changes. And the reason we can use C. elegans is it's a bacteriovore. That means the bacteria is both its food and it can also be its microbiota. So it gives us an unprecedented power to control genetically both the worm's genetic as well as the bacterial genetics and study their interaction. So over the last 10 years, we have been trying to understand this, and we use two different bacterial strains. One is the HT115, it's an E. coli strain, and they are very high in B12. So H, when you see H, it is high. And the OP50 is a low B12, so your O is the low B12, okay? Now the worms do not care. If you feed them either diet, they have normal life history, normal brood size, everything normal. However, serendipitously in the laboratory, you may come up with mutations which show altered life history traits, meaning that if, for example, they eat this neuromedin U receptor, it's a neuronal gene, when it eats this bacteria, it grows slower. On the other hand, if ALH6 bacteria grows on this, it becomes less fecund in the sense that it lays less number of eggs. And the opposite happens on the high B12 bacteria. We don't know whether that is B12 driven, but what we know is this gene, which is of serin-threonine kinase, which we've been working on for a while, when this is mutated, this pathway on a high B12 diet, and these worms live extremely long and they're extremely stress tolerant. So we started asking, how does this worm live longer on a B12 rich diet but not on the OP50 low B12 diet? And this constitutes a diet gene pairing which we are characterizing. So before I tell you about this story, I have to tell you the biochemistry 101 of vitamin B12. So vitamin B12 is an immensely complex metabolite, and I think it takes some 37 genes to synthesize vitamin B12. But vitamin B12 is used by only 2 enzymes in the entire body, entire biochemistry, only 2 enzymes. And the first enzyme is methionine synthase, which in C. elegans is called METR1. Methionine synthase is an important gene because in a B12 dependent manner, it converts homocysteine to methionine, and then methionine through SAMS1 is converted to SAM. So S-adenosyl methionine, which is a primary methyl donor in the cell. So all methylation reactions are governed by this methylation, and then it is replenished back to homocysteine. This homocysteine can either go round and round or enter the transsulfuration pathway, which is important for redox balance in the cell. It can also go down into the propionyl CoA, which is the main degradation pathway for odd chain fatty acids. So if you have a 3-carbon fatty acid which is from odd chain, the last product, it needs B12 to degrade it. And where does it go? It makes succinyl CoA. And you remember where succinyl CoA goes? It's in the mitochondria. So this whole path is in the mitochondria. and succinyl CoA enters the TCA cycle. So we will come back to this in a while. So over the last 10 years, as I was telling you, we've been extensively studying this serine threonine kinase gene, and what we know now is that when the worms have a mutant of this FLR4 kinase and it is fed on high B12 diet, the metabolism of one carbon cycle goes up. This leads to biosynthesis of serotonin. Serotonin binds to its downstream receptor, which sits on an interneuron, and then this interneuron secretes a neuropeptide, which is a glycoprotein hormone, that binds to FSHR1, which is its receptor in the intestine, and through phase transition of a SARM domain containing protein, activates a downstream cytoprotective pathway, which takes care of systemic aging in the intestine. And that's why these worms, when grown on high B12 diet, have higher lifespan and health. So this was, we have some more stories coming out on this. However, while we were working on this, we realized that we do not know what is the exact partner, ortholog of FLR4 in humans, because there is a very nematode specific gene, and we could not figure out which is the human ortholog. Otherwise, we could have been, in a great position. So we started looking at other genes, which, if it has a mutation, will phenocopy or emulate the phenotypes of FLR4. And luckily for us, we found one gene which is highly conserved, and it's called Richter. Richter is part of the TORC-C2 complex. I'm going to come to that in a while. And I'm going to show you assays, two assays that I'm going to talk about today. One is a stress tolerance assay there, and the other is a lifespan assay. So lifespan, if you have to do in the laboratory, you plot the survival, and this is on number of days. And as I promised, the worms which are wild type only live average of 17 days. That's what you see. But the wild types do not care. If you feed them high B12, low B12, they have normal life history traits. However, when the Richter worms are fed OP50 or low B12 diet, they have normal lifespan. You put them on high B12 diet, you see their lifespan is phenomenally increased, right? That's great. And this also reflects in their stress tolerance. So here we just put the worms on salt, a lot of salt, kept them for 10 minutes, then recovered them without salt. And you see the kinetics here. This is recovery, percentage recovery. The percentage recovery doesn't change if the wild type or the Richter worms are grown on these bacteria, but just look at the green bar. On high B12 diet, they are extremely long-lived. Okay? So in our assays, in everybody's assays, lifespan is most potently connected to stress tolerance. So from now on, I'm just going to show you one graph. And you have to believe me, if I show you that there is stress tolerance, it means they're also long-lived. So let me tell you a little bit about what Richter does. So Richter is a component of the TORC-C2 complex. So you have learned that the mechanistic target of target of rapamycin exists in two distinct complexes which have distinct functions, some overlapping functions that we know of. And TORC-C2 has Richter, whereas TORC-C1 has Raptor. So even if you do not have specific inhibitors for TORC-C2, which has prevented our understanding of this complex, you can knock down Richter, and that will kill the complex. The complex will dissociate. And this is important because you look at the orthology across the animal kingdom, and it's highly conserved. So it makes sense for us to go for this gene and understand whether this constitutes a diet-gene interaction. But before this, I have to convince you, right, that this is B12 dependent. So what experiment would you do? So you simply do this experiment where you take the low B12 diet, correct, where there is no change in the stress tolerance, and you gradually increase B12 by adding it to the media. And you see in a concentration dependent manner, you get back the stress tolerance. Just for the principle, you get back longevity. So that means that this is the Richter worms are sensitive to B12 and can better use B12 to promote a longer life, correct? Now let us go to the biochemistry and the signaling aspect of it. So I told you that B12 is required by two enzymes in this whole pathway. One in the 1-carbon cycle, where B12 uses homocysteine, it takes methyl group from here, converts it to methionine, and the other one is in the mitochondria, where it converts methyl manonyl CoA to succinyl CoA. So what I told you, I also proved by using metabolomic analysis that the flux through this pathway increases rapidly on high B12 diet, only on the mutant. And now what I did is, not me, my students, knock down these genes, okay, by simple RNAi. You knock down this gene or any of the genes of this pathway, and you see that the green line, which is the mutant on high B12 diet, do not have any stress tolerance. Similarly, whether you are knocking down the methionine synthase or the reductase, which is associated with that reaction. So essentially saying that this pathway is engaged, the metabolites through this pathway increases, and if you stop the flow of metabolites into this pathway, you prevent the increased longevity of the Richter mutants, even if there is high B12. So the next thing that we asked is that this succinyl CoA that I just talked about is converted to succinate in the TCA cycle, correct? And you all know that that is brought about by succinyl CoA synthetase, which in C. elegans has two isoforms. One, an ATP dependent, and the other one is GTP dependent. So we knocked down these isoforms and we asked, if we prevent the conversion of succinyl CoA to succinate, do we abrogate the phenotype? And the answer is yes. Whether you knock down the conversion through SUCA1 or SUCG1, you completely suppress the stress tolerance and longevity of the Richter worms on high B12 diet. Again saying that this pathway is engaged, and succinate may be the terminal metabolite. So we measured succinate in these worms, and lo and behold, in a B12 dependent manner, the succinate is much higher in these worms. So essentially, succinate is engaging a pathway which promotes longevity. Now, if succinate is the terminal nucleotide, I have to prove, right, it is a terminal nucleotide which is affecting it. So what we did is we knocked down SUCA1, SUCG1, stopped succinate conversion to, from succinyl CoA. It's suppressed. Now you add back succinate. It's an easy experiment. Just put it on the plate and let the worms feed on it. And you see that the worms now become stress tolerant again and again started living long. Saying that this pathway is engaging, succinate is upregulated, and succinate is required for the stress tolerance and longevity in the mutant. So the next obvious question is, what does succinate do in the mitochondria, right? So if succinate has increased in the mitochondria, it can also change mitochondrial dynamics. So let us look at how textbook talks about how the mitochondria looks like. So the mitochondria can exist either as fragmented into small pieces through a process called fission, which requires DRP1. It's an enzyme that squeezes the mitochondrial at the center and breaks it apart. Or you need mitofusions which will fuse the mitochondria and make a tubular network. Okay? So we asked whether this fusion-fission events are important for the longevity of the Richter mutant, because as I told you, maybe my succinate is engaging this machinery. So for that we started looking at mitochondria, and this mitochondria doesn't look anywhere near what you see in the textbook. So this is one muscle cell of C. elegans magnified many, many times. And if you see there, the wild type worms, and this is using GFP, which is targeted to mitochondria, you see that the mitochondria essentially fused when the wild type is grown on any bacteria, whether it is high or B12, low B12. The Richter mutant on low B12 bacteria behaves very similar to the wild type because they have all fused network. On the other hand, look at the mitochondria of the mutant on high B12 drive. They are highly fragmented. So you can use imagery to quantify this, and this comes out as increase in circularity. So your number of circles increase. That's why you see that on the last one, which is the Richter on high B12 diet, they have high circularity but a decreased aspect ratio. So this is, for every experiment we have done this analysis and measured statistically whether the differences are significant or not. But for the next slide onwards, I'm going to cut down the statistics. So just look at the figures. Now, okay. Now important fact is that, is this transfer into a fragmented mitochondria important? So we prevented fragmentation of mitochondria by knocking down DRP1. And you see that the mitochondria, when it cannot fragment, it cannot also support the increased stress tolerance and longevity, meaning that the succinate there was inducing mitochondrial fragmentation and promoting longevity. Importantly, if you now prevent the mitochondrial fragmentation, even your high B12 diet doesn't have any effect, right? So the high B12 diet was essentially fragmenting the mitochondria, and that was possibly going through the succinate. So to prove that, first what we did is we stopped the metabolites from flowing, either at the top by METR1 or methionine synthase knockdown, or MMCM1 at the bottom. And you see that in both cases, the fragmented mitochondria became fused. And you know that fused mitochondria does not promote longevity. But very importantly, you see that when we prevent the conversion, the mitochondria in the middle becomes fused. But now you add simply succinate to the system, you fragment the mitochondria. Again saying that it is the succinate which is coming through this pathway, which is fragmenting the mitochondria, and as a result, these worms are becoming long-lived. So what does fragmented mitochondria do? We know that fragmented mitochondria cannot promote longevity because they are not efficient in generating energy. So that was contrary to our understanding. So we asked whether mitochondria fission does something more important, which is activate mitophagy, the degradation of the mitochondria which are deteriorating, and using the autophagosome machinery to fuse it with the lysosome and clear it and regenerate raw material for the cells to grow. So for that, we looked at the pink and parking pathway, which are classical autophagy, mitophagy pathway genes. And in both cases, when you knock down PINK1 or PDR1, which is parking in C. elegans, you see complete suppression of the stress tolerance and longevity, saying that the Richter mutants engage the mitophagy pathway. We also looked at mitophagy by imaging in a very interesting way, but I'm not going to go into that. And we show that the mitophagy in the worms which are grown on high B12 diet, here the mitophagy, lower value means higher mitophagy. You see that they are high on mitophagy. On the other hand, if these worms are grown on low B12 diet, they are low on mitophagy. So this told us that maybe the fission of the mitochondria is activating the mitophagy pathway. So how do you prove that? You prevent the fission of the mitochondria. You don't allow the mitochondria to break. And that's the experiment we did. And we see that when the mitochondria is prevented from breakage by simply knocking down DRP1, the mitophagy decreases, right? So the value increases means mitophagy decreases. So in total, it tells that in the Richter mutants fed high B12 diet, succinate induces mitochondrial fission, and this broken mitochondria activates mitophagy. But how does that matter, right? How does activating mitophagy help the worms
Dr. Arnab,
live long?
can you please point
One slide, last. So we did a very interesting experiment where we show that as the worms grow older, the wild type worms have normal mitochondria fused, but as the worms grow older, the mitochondria starts fragmenting, which is not good. And that's what we know in literature, that as we grow older, the mitochondria fragments. But look at the Richter worms. The Richter worms have much better mitochondrial structure at 10 days, which is very old for the worms. And look at the mitophagy. Just by breaking the mitochondria very early in their life, these worms have maintained a consistently high mitophagy to clear away the defective mitochondria when the worms become old. So this brought us to this model, and I have already told you what happens, that in the Richter mutant worms, the flux through this one carbon cycle increases, leads to succinate production, which fragments mitochondria, induces mitophagy, and increases stress tolerance. On the other hand, this early mitochondrial fragmentation is helpful because in the wild type worms where green, the mitochondrial fragmentation increases with age. In the Richter mutant, the mitochondrial fragmentation decreases with age and they have tubular mitochondria. On the other hand, the mitophagy increases. And that's how these worms can engage a metabolic pathway in order to live longer. So this was work which was done by Simran Motwani, who has now graduated, and by Soumya Bhandari, who is now going for her PhD in McGill University. And I thank all my lab members, my fantastic collaborators, and funding agencies for their support. And I thank you for listening to me attentively.
Thank you, Dr. Arnab. It was a nice talk. We have already overshot the time. So one quick question from the audience.
Very nice talk. I just want to know, in Richter knockdown background, what about the AMPK mediated energy homeostasis in this model?
I didn't get.
In the Richter knockdown background, AMP kinase is also affected.
Yes, very
so
good
in that
question.
connection, how that energy homeostasis links to lifespan?
It's a very, very interesting question. So in the Richter knockdown worms, the AMPK is higher. So that's another project we are trying to understand. How, if this is a general nutrient deprivation because of, breakdown of mitochondria that is activating the AMP kinase. So we are looking at the signaling. This is only a, more cell biology, mitochondrial biology paper. We are writing a manuscript where we are looking at the signal transduction aspect and how lipid remodeling, which is regulated by Richter, that activates these pathways, including AMPK and P38 MAP kinase. Yeah.
Again, that mitochondrial fission also linked to the cell survival also maybe critically you have to answer, I think.
Maybe, yeah.
Thank you.
Thank you.
Nice talk.
Sir, you have mentioned about vitamin B12. So why can't we use probiotic lactic acid bacterium directly into the gut of the C. elegans, or?
It can be done, but it's, so I'm just telling you a very small part of the story. So we have also genetically modified the bacteria to show that it is B12 and not any other metabolite. Because here we are showing supplying the metabolite, right? But we make a B12 deficient bacteria, and we looked at the bacterial one carbon cycle to see how that affects. So putting a lactobacillus will have so many different metabolites that we will not be able to know exactly which metabolite. It could be B12 as well as many other things.
And another one, have you mentioned gut microbiota of C. elegans have been studied, or what, sir?
Pardon?
Gut microbiota of C. elegans.
So the natural gut microbiota of C. elegans is highly diverse and extremely consistent across different areas where it is. So we don't get C. elegans in India. So mostly, in European countries you get. But that gut microbiota is amazingly conserved. So they have a way to secure the diversity of their gut microbiota across the European continent. Yes, but we use monoculture because that gives us, it's much easier to understand the biology behind it. Thank you.
Wind up this. Thank you, Dr. Arnab. Wonderful talk.